When people see the word bacteria, they probably think about all the diseases they cause, all the times you got food poisoning. And they probably think that bacteria are the scum of the earth, just pointless little prokaryotes crawling around in the corners of our house. Well, bacteria are so much more than that. We choose to think of them as highly efficient and very useful factories. Because bacteria have a lot in common with factories. They have a way to take in raw material and spew out products through their cell wall. And inside these factories there is a master cookbook containing all the instructions for every single product made. That's the DNA. Now there's only limited numbers of copies of the cookbook, so of course the bacteria makes photocopies, which are the mRNAs. And these are spread out on the factory floor, the cell soma, where we have tools and workers, proteins and enzymes, that together make the products, which are mainly protein. And of course, there will be some source of quick energy on the factory floor to get the workers through their 24-hour shifts. Bacteria simply never sleeps. But there's one big difference between bacteria and real-world factories. That's this. You would never see a real-world factory self-replicate, but living cells do. If you can just convince one of these self-replicating factories to produce what you want in the way that you want it, you'd soon enough have a whole army of factories, and you could go on vacation to Hawaii. So if you're an engineer and you come up with a new cool pathway to produce whatever invention you've made, you would just need to write an instruction book and plant it inside a factory, and they will start producing your product for you. In the case of bacteria, that means making circular DNA constructs and transforming cells using this. And this sounds really easy, but in reality it's not so simple. We need to remember that we write this instruction book in the language of DNA, and even though we know quite a lot about this language, it's very easy to make grammatic mistakes or to misspell things, and your product will come out faulty, and your cell won't do what you want it to. Also, the cells are preoccupied with replication. That's their main purpose, right? So they have no actual interest in producing what you want. And the process of having bacteria take up your DNA can take weeks or months, and the design process is really slow because you need to troubleshoot with living bacteria. So a more clever engineer's approach is to crush the factory, steal the tools, and test your circuit outside the bacteria. And this is quite literally what we do in Richard Murray's biolab in the Keck building. But we use different words for it. We lyse the bacteria, we extract the tools, and we prototype our biomolecular circuits. And this gives us what we call a TXTL system. TX stands for transcription. That's the process of making photocopies, making mRNA from DNA. TL stands for translation, which is the process of taking mRNA and producing the products, the proteins. And we call this system TXTL because it has all the proteins and enzymes, the workers and the tools, that are necessary for doing these two processes. And that's all we need to actually test our instruction books. In the lab it looks like this. You take a tube with the extract, which is the crushed bacteria juice, and you mix it with a buffer containing amino acids and energy and some other things to set the conditions right. You mix those two together and blend it with the DNA that codes for your circuit. And you put that in a well, and if the DNA codes for a green fluorescent protein, the whole well will after a while turn green, and you can measure that using what we call a plate reader, which is basically just a camera. Green fluorescent protein is one of the main tools of synthetic biology. As we can't see normal proteins, it's very useful to have a protein that actually glows in the dark. It allows us to see what happens inside cells. and this was taken from a jellyfish and has been introduced to other organisms, and it's been used to make green fluorescent mice, green fluorescent monkeys, and kittens. So now I've given you the basics of what we do in the lab, how the TXTL system is used, and the rest of the time I will spend telling you about the circuit that I've made this summer. And the circuit design process has 3 first steps that I will talk about. The first one is design, the second is modeling, and the third one is the prototyping in TXTL. So first of all, let's look at a sub-circuit that is easy to understand. This contains 3 DNA pieces, as you can see, and at the bottom one we have something we recognize as DGFP. That's the green fluorescent protein. Now, for that to be produced, so we can measure it, we need to activate the promoter in front, which is called PGLNA. And this part is only activated if there's phosphorylized NR1 in the system. Now, what's that? Well, it's just a protein that has been phosphorylized by a kinase. And if you look at the top left DNA strand, we have NR1. So that can be produced by this DNA piece. But we also need the kinase that phosphorylizes it, and that's NR2. So those two combined will result in an activation of the PGLNA promoter, which then results in a production of DGFP. If you're into electrical engineering or computer engineering, you would recognize this as an AND gate. You can see in the truth table to the right, only when you have some amount of the 2 pieces NR1 and NR2, the production of GFP will start. The next step is to model this. So we use a model that takes into account all the chemical reactions between biomolecules and sets up ordinary differential equations to give us a model of how the system will behave. So what we see here is a graph where in the horizontal plane we have the concentration of DNA coding for NR1 and NR2, and on the z-axis we have RFU, which is relative fluorescence units, and it tells us how much GFP has been produced. The most important point here is that if we have 0 concentration of NR1 and NR2, we get no production of DGFP. But if we have a little bit of both, you can see we move up on the surface to the orange area, and that's where we have fluorescence. So this really looks like an AND gate, but let's test this out in TXTL and see what it really behaves like. And from TXTL we get graphs like this. On the x-axis we have time, so this is a 12-hour run. And on the y-axis we have the relative fluorescence units coming from DGFP. So we've been measuring for 12 hours how much fluorescence is in these wells. So we can look at the red dashed line where we have no NR1 or NR2 DNA in the system. And as we can see, there's some green fluorescent protein production even if we don't have these two present, which was not expected by us. So what we conclude from this is that the promoter is leaky, or perhaps that there's some NR1 and NR2 in the extract that we used to test this out, and we don't really know that. But we can at least see that the rate of production in the beginning, if we look at 100 minutes, it's fairly slow compared to the other curves. So let's compare it to when we add low amounts of NR1 and NR2. This is at 4 nanomolar. We see that the rate is a lot faster in the beginning, but it levels off fairly quickly. And if we move to medium concentrations of NR1 and NR2, the rate is even faster, and it reaches a higher endpoint. We also tested with too much of the NR1 and NR2, that would be 14 nanomolars, and we see the rate is really fast in the beginning, but it levels off faster than the other curves, because this much DNA uses up a lot of resources in the system. And remember how I showed you how we add an energy buffer? We only have limited amounts of energy in our system, so too much is too much. If we just look at the rates of this, we can plot something similar to what we modeled. So here once again we have the concentrations of DNA coding for NR1 and NR2 in the horizontal plane, and we have the peak rate of DGFP production on the z-axis. So if you look at the 0,0 point, that was the red dash curve in last graph, we see that we have a quite slow production. But if we introduce some NR1 and NR2, we get higher production rate. Well, the best case seems to be to have a little bit of NR1, but a lot of NR2. So this AND gate doesn't really behave as we modeled. There's some asymmetry here where one thing is more important than the other. And this is an example how TXTL can help us to gain knowledge about our DNA circuits before we try to implement them in bacteria. Just for reference, this is the full circuit that I've been working with this summer. This was the aim of the project, to complete this. In the bottom we see the AND gate that I've been talking about, and on top of that we've stacked a few additional layers so that this circuit can be controlled with the inducer molecules ATC and IPTG. And this allows us to control the circuit even if it's inside bacteria. We can expose the bacteria to these two chemicals and it will respond accordingly, which is really useful. Now I've prototyped all these different sub-circuits, but the next step would be to compile the whole thing and start trying to implement it in bacteria. And you see this circuit also follows a truth table, which is just an inverted AND gate. It's a NOR gate. Well, 10 weeks is not a long time when it comes to designing biocircuits, and this project still has a bit left before it can be implemented in bacteria. But by using the clever engineer's approach of prototyping before you start building in bacteria, we can speed up the process of creating engineered life forms. And this is really important because biomolecular circuits have a huge potential in many fields. If we can get better control over life forms, we could have them produce biofuels from waste and plant products. And we could substitute a lot of chemical production by having living cells produce the enzymes to catalyze chemical reactions, for example, to make pharmaceuticals. And why not monitor the environment for toxin spills? And you could even clean up maybe oil spills by having algae that chew up things that doesn't break down naturally in nature. I would like to finish off by thanking my mentor Richard Murray, my co-mentor Claire and Vipul, my co-surfers Tiffany and Pukit, who I've been working with the whole summer, and everyone else in the Biocircus lab for all the advice we've gotten and all the fun we had. Thank you. Now it's time for some bonus slides. These are things that didn't make it to the final presentation because they take a little bit of time to explain, but they're fun, so let's take a look anyway. We'll start out by going back to the main circuit, and here we see two pieces that are called anti-1 and anti-2, and they have these red repressor lines going to the at1 and at2 parts of the next constructs. And we wanted to test out how this works. So we designed little sub-circuits, or they're just really test constructs, using 4 DNA pieces, as you can see here. And the idea is that the anti-1 is supposed to stop the production of the GFP if there's a little at1 part in front of it. And the way this happens is, for example, if we have A66, the attenuated 1 mRNA will be produced. And this doesn't translate into any protein, but as an mRNA piece, it has the property that it's able to bind to the attenuated region of another mRNA piece. So that means all the mRNAs that are produced from A59 that contain the GFP, they will be sequestered by the anti-1 because it binds to it and makes it possible for the protein production to happen, the translation. And the same thing with anti-2 and attenuated 2. Now, partly what we wanted to do here is to test so that anti-2 doesn't stop the production of attenuated 1, or vice versa. So the first thing we did was to model this, and in the graph below each of the circuit diagrams you see the modeling results. The black line is with none of the 2 antisense, just the construct on its own, and you can see it produces some fluorescence. If we start by looking at the attenuated 1, so that's the left side of this page. And the orange line is then when you add the antisense 1 DNA that codes for that. And you can see the orange line drops fairly low, much lower than the original line. The blue line is when we add the same amount of antisense 2, and instead we see the production go up. If anything, this shows that antisense 2 doesn't stop attenuated 1. But why does it go up so much? Well, we believe this is because when we produce a lot of mRNA in the system, there are always enzymes to try to break these down, because they're not supposed to remain in the cell. So if we produce a lot of mRNA that is not translated into protein, just junk mRNA, we will keep these enzymes busy with chewing up the junk, and our non-junk, the DGFP mRNA, will remain longer in the system and result in a higher fluorescence. And testing this, if we go down to the bottom row on the left side, we see we tested this in TXTL with the constructs that are above, and we see the black line is where there's no antisense 1 and no antisense 2, and if we add antisense 1, the production goes down just a little bit, and if we add antisense 2, the production actually goes up. So it looks just like we modeled, but the difference between the curves is not the same. If you look on the right side, you have all the attenuator 2 plots, and you have the modeling. It shows the same behavior, but the orange and the blue line are swapped around, right? So introducing antisense 1 to attenuator 2 just increases the production, while if you add antisense 2 to attenuator 2, just like we expected, the expression goes down heavily. And below that you see the TXTL run of the same thing. So this was just another example of how we can prototype sub-circuit parts and test the functionality of parts in TXTL before we start doing in vivo work with bacteria.